So students, in our previous video, we had studied the chapter till now. Now we are going to start with Mendel's experiments. Now, first of all, who was Mendel? So Grigel Mendel was the first scientist to make a systematic study of patterns of inheritance which involved the transfer of characteristics from parents to progeny. Mendel is also known as the father of genetics. Mendel chose pea plants for studying inheritance because pea plants had a number of clear-cut differences which were easy to tell apart. Now, first of all, before moving ahead, we need to understand some rules of inheritance of traits. So, the rules of inheritance of such traits in human beings are related to the fact that both the father and the mother contribute practically equal amounts of genetic material to the child. This means that each trait can be influenced by both paternal and maternal DNA. Thus, for each trait, there will be two versions in each child. And that is which was kind of studied by Mendel. Mendel used a number of contrasting visible characteristics of garden peas that can be round or wrinkled seeds, tall or short plants, white or violet flowers, and so on. Another reason for choosing pea plants was that they were self-pollinating. Also, Mendel chose pea plants because many generations of pea plants can be produced in a comparatively short time span, and their study is much simpler than that of animals. What happens when pea plants showing two different characteristics rather than just one are bred with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and roundness seeds are thus dominant traits. But what happens when these F1 progeny, that is the first filial generation, are used to generate F2 progeny, that is the second filial generation, by self-pollination? A Mendelian experiment will find that some F2 progeny are tall plants with round seeds and some were short plants with wrinkled seeds. However, there would also be some F2 progeny that showed new mixtures. Some of them would be tall but have wrinkled seeds while others would be short, and all of these techniques can be studied through the Punnett square method. Now we'll quickly move on to the laws given by Mendel, that is the laws of genetics. First, law of segregation. Mendel's first law of inheritance, or the law of segregation, suggests that the characteristics of an organism are determined by internal factors, that is now known as genes or DNA, which occur in pairs. Only one such factor can be represented in a single gamete. Some of the key points of this law of segregation were: each gamete acquires one of the two alleles as chromosomes separate into different gametes during meiosis. Heterozygotes, which possess one dominant and one recessive allele, can receive each allele from either parent and will look identical to homozygous dominant individuals. The law of segregation supports Mendel's observed 3:1 phenotypic ratio. Mendel proposed the law of segregation after observing that pea plant with two different traits produced offspring that all expressed the dominant trait, but the following generation expressed the dominant and recessive traits in a 3:1 ratio. Mendel's second law of inheritance, or law of independent assortment, states that in the inheritance of more than one pair of traits in a cross simultaneously, the factors responsible for each pair of traits are contributed independently to the gametes. Now, to simplify it, let's study this from the beginning. To make an accurate prediction, we need to know whether the two genes are inherited independently or not. That is, we need to know whether they ignore one another when they are resorted into gametes, or whether they stick together and get inherited as a unit. When Gregor John Mendel asked this question, he found that the different genes were inherited independently of one another, following what's called the law of independent assortment. In this article, we'll take a closer look at the law of independent assortment and how it is used to make predictions. Now that we have studied how the transmission of the traits takes place, we need to study that what is the process of it. The characteristics or traits of parents are transmitted to their offspring through genes present on the chromosomes during the process of sexual reproduction. Genes work in pairs. For every trait, there is a pair of genes, that is, one dominant and one recessive. Each parent passes only one of the two genes of the pair of each characteristic. Though the offspring inherits two genes for each trait from its parents, but the trait shown by the offspring depends on which inherited gene is dominant of the two. So now let's get started with what are genes. When a male gamete fuses with a female gamete during fertilization, they make a new cell which is known as zygote with a full set of genes. That is 2N. Now this zygote grows and develops to form a new organism having characteristics or traits from both the parents which it has inherited through genes. The two genes responsible for a particular characteristics are always present on the corresponding positions of the pair of chromosomes, which are also known as the homologous chromosomes or the sister chromatids. Now students, we have already discussed the idea that the two sexes participating in the sexual reproduction must be somehow different from each other for a number of reasons. Now, how is the sex of a newborn individual determined? Different species use very different strategies for this. Some rely entirely on environmental cues. Thus, in some animals, the temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be a male or a female. In other animals such as snails, individuals can change sex, indicating that sex is not genetically determined. However, in human beings, the sex of the individual is largely genetically determined. In other words, the genes inherited from our parents decide whether we will be boy or a girl. But so far, we have assumed that similar gene sets are inherited from both the parents. If that is the case, how can the genetic inheritance determine sex? The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have a maternal and a paternal copy, and we have 22 such pairs, that is 22 autosomal chromosomes. But one pair called the sex chromosome is odd in not always being a perfect pair. Women have a perfect pair of sex chromosomes, both called X, but men have a mismatched pair in which one is a normal sized X while the other is a short one called Y. So women are XX and men are XY. So hence all children will inherit an X chromosome from their mother regardless of whether they are boys or girls. Thus the sex of the children will be determined by what they inherit from their father. A child who inherits an X chromosome from her father will be a girl, and one who inherits a Y chromosome from him will be a boy. And thus by this technique one can determine the sex of a newborn baby.